The soybeans that are grown in Minnesota are not very diverse. They trace back to probably 10 to 15 parents that were originally introduced from China. So compared to a lot of other crops, the soybean genome is not very diverse. We need to create new genetic variation because when new diseases or insects or new environmental challenges come along, we want to have diversity so that we can select new genotypes and varieties that will be able to deal with those changes that will come about.
So long term, I think it's an important initiative to try to create new genetic resources in which to use for future breeding applications. I guess you could say we have two projects in this area. We're involved in defining the genetic changes that are underlying some of the differences in seed composition that we're seeing in the fast neutron mutant population that's been developed here at the University of Minnesota. You treat the seed with some sort of mutagen, be it chemical or some sort of particle, and you hope to get a new type of DNA variant that's quite useful and novel.
The Christmas tree mutant is a plant that we created using fast neutron mutagenesis. The fast neutrons are a kind of atomic energy that causes the DNA in the soybean genome to be split apart and then create gaps or deletions in that DNA. It has a large deletion of several thousand base pairs, and the plant has a very unique phenotype. That is, instead of being the normal kind of bushy soybean plant, it has more of a Christmas tree shape with a kind of a pointed top and a fairly narrow profile, which we believe might have some opportunity to be utilized in the narrow row production that we tend to recommend for the northern part of Minnesota.
It's very early, but we have some preliminary data that tells us that this could potentially be a very useful plant.
This is an example of the type of work that is a long-term undertaking. It gives us an opportunity to understand more about the basics of soybeans, but also take that basic information and then apply it to a breeding program that would eventually develop varieties that could be used by Minnesota farmers.